Technical Veterinary Learning TVL channel, founded and produced by Professor Ahmed Mamdouh El Sharif, Professor of Epidemiology and Infectious Diseases of Animals, the ex-head of Department of Veterinary Medicine, the ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University. We will speak today about an important infectious disease of all species of animals that take several names as the following: Dermatophilosis, exudative dermatitis, lumpy wool of sheep, cutaneous streptotheratosis, Syncopo disease of cattle. Dermatophilosis is a worldwide contagious bacterial skin infection affecting all animal species and man. It is most common in immunosuppressed animals. It is most common in animals chronically exposed to wet weather. It is the most common disease in young. Historical view: Van Skagum in Congo Republic at 1915 write the first scientific report about dermatophilosis. Epidemiology: pathogenic risk factor. Pathogenic risk factor. There are two pathogenic risk factors for dermatophilosis. The main one is the Dermatophilus congolensis. The secondary factor is damage to the skin by different causes to fulfill infection. Scientific classification of the main factor of dermatophilosis. It belongs to domain bacteria, phylum Actinomycetota, class Actinomyceta, order Micrococcales, family Dermatophyllaceae, genus Dermatophilus, and the species is Dermatophilus congolensis. The morphology of Dermatophilus congolensis. It is a facultative anaerobic actinomycete. It has two morphological forms, filamentous hyphae and motile zoospores, as it will be shown in the following slides. Morphology of Dermatophilus congolensis. The hyphae are characterized by branching filamentous with 1 up to 5 micrometer in diameter that ultimately fragmented by both transverse and longitudinal separation into packets of coccoid cells. These coccoid cells mature into flagellated ovoid zoospores with 0.6 up to 1 micrometer in diameter. With microscope, you can observe the characteristic tram-car line, Dermatophilus congolensis colonies, together with gram-positive thin filamentous and coccoid forms. Dermatophilus congolensis under microscope appear as in this slide is a gram-positive microaerophilic bacteria showing filamentous hyphae and motile zoospores. Filamentous mycelial-like growth of Dermatophilus congolensis showing transverse separation or tram-car line. Filamentous mycelial-like growth of Dermatophilus congolensis showing the tram-car line as appear in this slide. Dermatophilus congolensis is a gram-positive microaerophilic bacteria, motile zoospores, and filamentous mycelial-like growth showing transfer separation or what's called tram car-like. Tram car-like of filamentous mycelial-like growth of Dermatophilosis congruensis that take gram-positive stain and appear like that as in this slide under the microscope. Cultivation of Dermatophilosis congruensis. Dermatophilosis congruensis is a carboxyphilic germ by means needs carbon dioxide to probably grow on laboratory media. The germ grows well on sheep blood enriched agarose medium. Media must be incubated at 37 centigrade and in 5 to 10% carbon dioxide atmosphere. Beta hemolysis can be seen around the colonies. This will be described well in the following slides. Dermatophilosis congruensis. Colonies A and P appear 24 to 48 hours, are small with about a 1 millimeter diameter and with gray-yellow color, as appear in this slide. Dermatophilosis congruensis colonies host 3 to 4 days. Isolated bacterial colonies reach to 3 millimeter in diameter and have a rough surface and yellow-golden pigmentation. Dermatophilosis congruensis germ does not live well in the normal environment by means that outside the body. It is susceptible to external influence of pH and moisture fluctuation. Geographical distribution of dermatophilosis. Geographical distribution of dermatophilosis. It causes epidemics in tropical and subtropical areas with considerable economic losses. In Africa, prevalence of Dermatophilosis congruensis infection in cattle reached to about 15%. Infection rate reaches to 100% in some herds during the time of peak seasonal prevalence. Geographical distribution. Dermatophilosis reported throughout the world. Sporadic cases reported in temperate areas. The incidence varies from 10 to 66 in areas have shower cooling, which act as predisposing factors, and it acts a factor of skin destruction. High prevalence in sheep flux reported in high and medium rainfall areas, and significant clinical cases were reported. It has been reported in Australia, Canada, northern of United States, Scotland, New Zealand, and Africa. The World Geographical Distribution of Dermatophilosis Map 2022. Orange color demonstrated the infected areas, while yellow zones is questionable. Blue zones are free areas. Host risk factor of dermatophilosis. These are the host risk factors of dermatophilosis: cattle, sheep, goats, camels, deer, horses, donkeys, pigs, wildlife species, and dogs. Infection reported in all animals' ages that ranged between suckling weeks up to most age 1. In case of host risk factor, breed differences in susceptibility are also apparent, and genetic markers have been identified in cattle and sheep. In Africa, Nadama and Motro cattle breeds are also as native breed sheep are resistant, while also in Africa, Zebu, white Fulani, and European cattle breeds are susceptible to this disease. The susceptibility in cattle can be influenced by genetic selection. Merino sheep of strong or medium wool strains, open fleeced sheep, and those with low wax and high sunt content in their fleece are more prone to infection. Sources of infection. The outbreaks exist with minor active lesions on the both face and feet of carrier animals, and may also be present in haired areas on face, feet, and on the scrotum, as well as on the skin along the dorsal midline of the back in carrier lambs. Scabies will carried in hair and wool from healed lesions act as the major sources of infection. Subclinical carriage form of dermatophilosis is common in most flocks where minor lesions are most evident at the junction of hair and non-haired areas of nares, claws, and declaws. Transmission of infection. Natural cutaneous defense mechanisms. Number 1, stratum corneal barrier. Number 2, superficial wax layer produced by sebaceous glands. Number 3, on the body of sheep, the physical barrier of the wool itself. Predisposing factors for Dermatophilosis congilensis transmission. Number 1, it is not a highly invasive organism and does not normally pass later natural barrier of healthy skin that we described. On whole skin, feet and face skin barriers are easily and commonly broken by different abrasive terrain, such as wire boundaries between farms, thorny spiny forage, harsh feed stuffs. Number 3, there is a particular tendency for lesions to occur on the rump and back skin in females and males, probably due to introduction of infection through minor abrasions caused by mounting. Number 4, abrasions and direct infection from the claws during mounting resulted in lesions down to flanks of cattle. Other predisposing factors for transmission are other penetrating lesions due to ear tags or biting flies that resulted in minor lesions. Number 6, usage of periodic contaminated showers or continual misting to cool cattle during hot periods is a risky factor for infection in dairy herds. Number 7, intercurrent disease is also a risk factor, and in infected dairy herds, a higher incidence has been observed during first weeks of parturition in first calf heifers that suffered from endometritis or mastitis. Skin infection occur by direct contact and mechanical transmission. The direct contact, the main mode of transmission that occurs between different carriage lesions on the face of one animal to fleece or skin of another one, from feet of males to skin of females at mounting during breeding seasons. Mechanical transmission, reported by flies and ticks along the surface of shearing cuts and mediate infection during contaminated dipping. Resulted on lesions don't spread over the body, but provide a significant source of infection for others in free flux, especially when management system or climatic circumstances lead to a high degree of susceptibility. Number 3, dermatophilosis in Africa is often combined with demodicosis to produce Syncopo disease that is a more severe and often fatal combination. Also, mechanical transmission occur due to trauma to the skin produced by thorny bushes and the oxpecker, per Buphagus africanus can also initiate the lesions. Dermatophilus congolensis may infect these lesions and then transmitted mechanically by different feeding flies, which result in minor infection. Also, one way of skin infection methods, occurrence of dermatophilosis following to infection with contagious eczema. Transmission in case of equines occur through biting flies, ostomoxis, housefly are thought to act as a mechanical and carrier vectors of infection. Skin damage of equines may be occur due to trauma, ectoparasites, wetting from rainfall, or from frequent washing. Environmental and managemental risk factors in case of dermatophilosis. A prolonged and heavy rain seasons is sufficient to produce the disease. Attack rates reach up to 50%, especially if followed by warm and humid weather, which retard drying of fleece. Prolonged wetting of sheep fleece is a major risk factor that leads to emulsification of wax barrier and maceration of cutaneous surface with disruption of stratum corneum. Moisture and rains releases infective zoospores from carriage lesions which carried mechanically by flies that are attracted by wet wool. Moisture of fleece and positive chemotactic response to carbon dioxide that present at cutaneous surface in addition to motile zoospores are aided in movement of them. Incidence of all cutaneous dermatitis in sheep has been increased within the time elapsed between shearing and dabbing due to dabbing, jetting, or spraying of sheep for external parasites with contaminated solutions. Economic importance of dermatophilosis. Economic importance of dermatophilosis. Dermatophilosis causes about 17% of deaths and culling rates in cattle and sheep flocks. Decreased work ability of infected equines, as well as infected cattle. Reproductive failure due to vulva infection in all animal species. Male limbs infection will prevent surface mounting during heat seasons. Deaths of newly born animals due to starvation and due to dams under infection. Losses of meat and milk production due to deaths. Losses of high skin grading. 23% fall in the milk production of infected animals that shows mastitic infection. Economic importance in case of sheep and goats. Number 1. Loss of 30% of wool value and 40% losses of skin value. Extensive losses in lambs that spring lambing has to be abundant. Interference with shearing. Number 4, a very great increase in susceptibility to blowfly infestation. Economical importance in case of African cattle. Major bacteriological diseases cause deaths are dermatophilosis, brucellosis, and contagious bovine pleuropneumonia. Zoonotic implications: dermatophilosis, an occupational disease in human, present on hands of veterinarians, poachers, farmers working with infected animals. A picture of an infected leg, thumb, in a man with dermatophilosis. An infected hand, thumbs, in a man with dermatophilosis, where the arrows denote to the area of infection. An infected arm in a man with dermatophilosis lesions, where the area of infection shown here in this picture surrounded by black ring and the arrows denoted to the lesions. Dermatophilosis lesion in an infected hand in a man, where the arrows denote to the lesion. An infected leg in a man with multiple dermatophilosis lesion, showed here surrounded by yellow circles. Pathogenesis of dermatophilosis. Dermatophilus congruensis germs established the infection and multiplication of themselves in the epidermis of the skin by secondary factors such as trauma, ticks, rain, hair and wool wetting, humidity, warm temperature. The enzymatic virulence factors, adenase and lysosinase of Dermatophilus congruensis, help in rapid multiplication of the hyphae and massive infiltration with neutrophils, where repeated cycles of invasion will occur in the same place, leading to typical pyramidal shape escapes formation in case of cattle. Formed escapes separate from healed lesions with holding loosely in its place by hair or wool. In case of cattle, immune suppression that resulted from tick infestation promotes the spreading of skin lesions, while in sheep, huge cutaneous maceration that noted with prolonged flea sweating resulted in extensive lesions under the fleece. All of this will lead to infected escapes are the source of repeated and expanding invasion that reported until developing of immunity and healing of the lesion in the same animal and in the flux. Extensive suppuration and severe toxemia may occur due to secondary bacterial infection. Clinical picture. Clinical picture of dermatophilosis in sheep. Cutaneous lesions are usually not visible due to the size of wool that obscured them. Scabs should be palpated as hard masses at skin surface, which typically distributed irregularly over the dorsal midline with ribs, then spreading laterally and ventrally. These lesions give the disease name of lumpy wool disease. These crusts are circular, rough, up to 3 cm in thickness, distinctly pyramidal with concave face, often pigmented, and underlying skin is moist and reddened. Muzzle, face, ears, and scrotum of rams may also be involved. Health of diseased sheep is not affected unless skin lesions are definitely widespread. Huge mortalities recorded in young lambs with extensive skin lesions all over the body. Secondary pneumonia and skin blowfly meiosis in occasional cases due to affected organisms. Death of affected sheep due to occurrence of secondary bacterial diseases. This picture showing dermatophilosis lesions in a sheep on ears, eyelids, frontalis, nares, and muscles, where the arrows are potted. Dermatophilosis lesions in sheep as crusty and scaly lesions involve dorsal line and lateral sides of the body. Black arrows here denoted to the place of lesions. Lesions of dermatophilosis in a sheep as crusty and scaly lesions involve several areas of the body. Dermatophilosis lesions in a sheep as huge cutaneous maceration that notes it to involve several areas of the body. Dermatophilosis lesions in a sheep as scaly lesions involve several areas of the head. Yellow rings and arrows denoted to dermatophilosis lesions places in a sheep as matting wool lesions involve several areas of the neck. Matting wool lesions involve several areas of the neck where dermatophilosis lesions will be noted in a sheep. The arrow and ring here denoted to that lesion. Picture of dermatophilosis in goats. Lesions appear first on the lips and muzzle and then spread, possibly by biting, to the feet and the scrotum. They may extend to all parts of the body, especially the dorsal midline and inside the thighs. In some cases, lesions commence on the external ears. Heavy crustus formation may block the ear canal and the external nares. This head of an infected goat with dermatophilosis lesions on ears, eyelids, frontalis, nares, and muscles. Yellow and black arrows denote to that. This head of the goat denoted to dermatophilosis lesions as crusty and scaly lesions involve head, ears, and neck. Crusty and scaly lesions involve eyelids, muzzle, and ears in a goat infected with dermatophilosis. Crusty and scaly lesions involve the scrotum of an infected male goat with dermatophilosis. Clinical picture in cattle. In case of cattle, early lesions is a pustules and the hair over the infected site is erected and matted in tufts with greasy exudate, forming crumply crustus which are hard to remove. These develop two scapes which are greasy and fissures at flexion points, and finally two scapes that are hard, horny, and conflict. The scapes vary in color from creamy to brown, are 2 to 5 centimeters in diameter, and are often in such close opposition that they give the appearance of mosaic-like. In the early stage, the crusts are very tenacious and attempts to lift them, causing pain. Beneath the crusts, there is granulation tissue and some pus. In the latter stages, the dermatitis heals and the crusts separate from the skin are held in place by penetrating hairs but are easily removed. Lesions occur on the neck, body, the back of the udder, and may extend over the sides and down the legs and the ventral surface of the body. Commonly, they commence along the back from the withers to the rump and extended halfway down the rib cages. In some animals, the only side affected is the flexor aspect of the limb joints or the inguinal area or between the forelimbs. In young calves, infection commences on the muzzle, probably from contact with the infected udder or because of scaring by milk in bucket-fed calves, and may spread over the head and neck. This picture showed dermatophilosis lesions in a cattle with the scabs vary in color to brown, are 2 to 5 cm in diameter, and are often in such close apposition that they give the appearance of mosaic. Dermatophilosis lesions in a cattle shows scabs, and sites affected are the flexor aspect of the limb joints or inguinal areas or between the forearms. Dermatophilosis lesions shows scabs, and sites affected are the flexor aspect of the limb joints or inguinal areas at cattle, as well as appear distributed on the scrotum. A collection of scabs at the dorsal line of a calf body due to dermatophilosis. Bovine dermatophilosis, exudative crusty skin lesions at the pelvic area. Dermatophilosis lesions in a cattle shows scabs brown in color, are 2 to 5 cm in diameter, the close opposition that they give the appearance of sticky matted hair at the dorsal line. Dermatophilosis clinical picture in camels. In case of camels, dermatophilosis lesions are scaby lesions that occur on the neck, body, the back of the hump, and may extend over the sides and down to the legs and the ventral surface of the body. Dermatophilosis lesions as scabs appear at the neck of a camel. Dermatophilosis lesion after healing in the face of the camel under the ear. The black ring here denote to the place of that lesion. Dermatophilosis is scabby lesion in the abdomen of a camel. The yellow ring around dermatophilosis lesion in abdomen showed hairless brownish crusts with irregular sizes and pus exudation. Clinical picture of dermatophilosis in equines. Dermatophilosis lesions in equines characterized by the hairs are matted together over the lesion, and an extensive dermatitis produces a fine mat of hairs and debris just above the skin surface. If this hair is plucked, the entire structure are left off, leaving a characteristic ovoid, slightly bleeding skin areas. No pruritus, no irritation is apparent, although the sores are tender to the touch. Infection can appear on the head, beginning at the muzzle and spreading up to the face, to the eyes, and if sufficiently extensive may be accompanied by lacrimation and a profuse mucopurulent nasal discharges. Clinical picture of dermatophilosis in equines shows the following. In some horses, these lesions are confined to the lower limb with a few on the belly area. In very bad environmental conditions, the lesions may be widespread and cover virtually the whole of the back and sides. The lesions of dermatophilosis on the lower limbs are most common behind the pastern, around the coronet, and on the anterior aspect of hind cannon bones. If the underlying skin cracks, the horse can become very lame. This variable distribution of lesions may depend upon the existing skin trauma, which act as the secondary factor of appearance of this disease. A very clear picture of dermatophilosis at the pelvis area of a horse, showing matted hairs together over the lesion with an exudative dermatitis, giving a fine mat of hairs and debris just above the skin surface. The yellow ring surrounded the lesion, to be clear for you. The lower areas of donkey's limbs showing very clear dermatophilosis lesions, where matted hair together over the lesion with an exudative dermatitis gave a fine mat of hair and debris just above the skin. At the right forelimb, a yellow circular area showing you the lesion, while on the left limb there is marked yellow diagram around the lesion, to be clear for your eyes. A very clear dermatophilosis lesion taking approximately the whole entire body of the skin surface of an infected horse, where matted hair together over the lesion appear with an exudative dermatitis. A donkey in very environmental bad conditions showed dermatophilosis lesions widespread and cover virtually the whole of the back and sides. Matted hair together over dermatophilosis lesions at the pelvis area of an infected horse with an exudative dermatitis gave a fine mat of hair and debris just above the skin surface. Dermatophilosis lesions over large areas showing scaly allopathic areas almost at the entire body of an infected horse. Dermatophilosis lesions in an infected horse as hair are plucked and the entire structure may lift off, leaving the characteristic ovoid slightly bleeding skin area. Dermatophilosis lesions as hair are plucked and the entire structure may lift off, leaving a characteristic ovoid slightly bleeding skin area over the back and pelvis area of an infected horse. Dermatophilosis, large areas of scaly and matted hairs lesions at the entire neck and thorax area of an infected horse. An infected horse with Dermatophilosis congruences showed lesions configured to the upper part of the body with a few on the belly of matted hairs together. Matted hair together over the lesion with an exudative dermatitis gave a fine mat of hair and debris at large scale of skin surface and the tail of a diseased horse with dermatophilosis. A horse forehead showing matted hair together over the lesion with an exudative dermatitis. This horse is infected with dermatophilosis. Matted hair together over dermatophilosis lesions with an exudative dermatitis gave a fine mat of hair and debris at skin surface of neck, scapula, and thorax of a diseased horse. Thorax and belly areas of a diseased horse with dermatophilosis showing matted hair together with an exudative dermatitis. Dermatophilosis at skin surface of the base of the tail and pelvis area of a diseased horse with dermatophilosis showing matted hair together over the lesion area with an exudative dermatitis gave a fine mat of these hair with debris. A horse in very bad environmental conditions showed dermatophilosis lesions widespread and cover virtually the whole thorax area. Dermatophilosis is common in most flocks where minor lesions are most evident at the junction of haired and non-haired areas of claws and declaws of infected donkeys. Dermatophilosis lesions as hair are plugged and the entire structure may lift off, leaving a characteristic ovoid, slightly bleeding skin area at the lower limb of an infected donkey. Dermatophilosis clinical picture in dogs. Matted hair together over the lesion with an exudative dermatitis gave a fine mat of hair at the area of an infected dog with dermatophilosis. Dermatophilosis lesions in a dog shows scabs are often in such close a position at the areas of the two ears and muzzle and around the eyelids. Dermatophilosis lesions in a dog shows scabs and scales are often in such close a position at the area of the forelimb above the fingers. The black arrow and black ring here showing you the place of the lesions. This picture showing a face of dog and the rectangles in blue color showed dermatophilosis lesions with scabs and scales under the eyelids and at the frontalis. Laboratory diagnosis of dermatophilosis. Clinical samples that collected in case of dermatophilosis, scrapings or a biopsy section, and is much easier to isolate from an acute case than from chronic one. Polymyxin B sulfate can be used to suppress contaminants. Laboratory tests for clinical specimens collected from cases suspected to be infected with dermatophilosis. The following tests are applied for diagnosis of dermatophilosis. Direct impression smear to detect the Dermatophilus congruensis, hyphae, and motile spores. Fluorescent antibody technique, ELISA test, PCR, sequencing of the isolated strains, and immunoelectrophoresis. This microscopical field showing Dermatophilus congruensis, motile zoospore, and filamentous hyphae inside the infected skin tissues. Microscopical field showing filamentous mycelial-like growth of Dermatophilus congruensis with transverse separation inside cutaneous infected tissue with tram car-like shape. PCR of a specimen of an infected animals from number 3 line up to number 13 line with 500 base pair showing the positive of these clinical samples for presence of Dermatophilus congruensis. Phylogenetic analysis of Dermatophilus congruensis isolated from naturally infected cattle showing there are no difference in their isolated strains and all of them have the same phylogenetic tree with small differences between them. Post-mortem findings in case of Dermatophilosis. The main post-mortem findings in case of Dermatophilosis are extensive dermatitis in dead animals, sometimes a secondary pneumonia, bowel flies, and other counter diseases. This picture showing leukocytes infiltration in the different cutaneous layers due to dermatitis from infection with Dermatophilus congruensis. Differential diagnosis. Dermatophilosis have to be differentiated from the following 6 diseases: ringworm, staphylococcal dermatitis, staphylococcal folliculitis, scabies, pediculosis, fleece rot in sheep. Treatment of dermatophilosis. Treatment of dermatophilosis in sheep, including bactericidal DEPs, procaine penicillin with 70,000 international units per kilogram, combined with streptomycin with 70 mg per kilogram. Long-acting tetracycline at 20 mg per kilogram body weight. Erythromycin at 10 mg per kilogram body weight. Number 5, combination of lincomycin and spectinomycin at a dose of 5 mg per kilogram body weight. Number 6, the usual strategy is treat 8 weeks prior to shearing, so there is time for the lesions to heal and shearing to occur without interference from active lesions. Number 7, antibiotic treatment at a high dose for a single treatment is effective in reducing the proportion of active lesions in an affected flux. Treatment of dermatophilosis in cattle at temperate areas. Number 1, tetracyclines 5 mg per kilogram body weight repeated weekly as required is recommended. Number 2, long-acting tetracycline 20 mg per kilogram body weight in one injection is reported to give an excellent result in affected cattle. Parenteral procaine penicillin 22,000 international unit per kilogram body weight daily for 3 days is also reported as efficacious. Treatment of dermatophilosis infection in cattle at tropical areas and associated with tick infestation. No completely satisfactory treatment in these herds with extensive involvement, or those being constantly reinfected or exposed to predisposing causes. Better results are obtained during dry weather and in dry climates. Parenteral treatment with antibiotics can be used and should be used in conjunction with acaricides when ticks are present. Treatment of dermatophilosis in equines. Number 1, topical therapy is most used in equines coupled with removal from whatever is causing prolonged wetness of the skin. Number 2, scabies can be removed by grooming, that done under sedation. Number 3, lesions treated topically daily with bovidine iodine or chlorhexidine cream until the lesions are healing. Treatment of dermatophilosis in equines, including also benzyl peroxide, which has keratolytic, antibacterial, and follicular flushing properties, and is reported to be effective in therapy when applied topically at a concentration of 2.5%. Number 5, diseases associated with actinomyces species, actinobacillus species, nocardia species, and dermatophilus species, as severe cases, can be treated daily for 3 days with penicillin at 20,000 international unit per kilogram intramuscularly. Control of dermatophilosis. Control of dermatophilosis including the following. Number 1, avoid all predisposing factors. Number 2, isolation of infected animals from sound one. Number 3, usage of separate grooming utensil per animals. Number 4, shearing and dabbing of infected animals at the last trip. Number 5, insecticidal dabbing should contain a bactericidal treatment. Number 6, closed yards or factors that promote face-to-skin contact. The last step in controlling of dermatophilosis, where dabbing immediately after shearing is a great risk factor, the severity of infection can be reduced by delaying dabbing. For example, from the day of shearing to the 10th day post that, or by dabbing in zinc sulfate immediately following shearing with later dabbing in an insecticide. We're keeping you on the right track, so please subscribe the channel to reach to the most newest knowledge about the epidemiology and different infectious diseases of different species of animals. See you later.